Hello everyone, and welcome to today's live broadcast, Insights into Virus Replication, the Role of Co-opted Host Proteins and Lipids, presented by Dr. Peter Nagy, a professor in the Department of Plant Pathology at University of Kentucky. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're excited to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on that green Q&A button located on the lower left of your presentation window. Type your questions into the box that appear on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you are viewing this presentation in a slide window. To enlarge that window, just click on the screen icon on the lower right. If you have any trouble seeing or hearing this presentation, please click on the support button at the top right of the presentation window, or use the Q&A button and let us know you're having a problem. Good news, this presentation is educational and thus offers continuing educational credits. Click on the button in the bottom left-hand corner and follow the process for obtaining your credits. Without further ado, please join me in welcoming Dr. Peter Nagy. I will now turn the presentation over to him. Welcome, sir.
Everyone, today I'm going to talk about how viruses utilizes the cell, how they are using the cell resources to make a lot of new progenies, and how the infection goes on. So I'm talking about RNA viruses. These viruses have RNA genome. And so they don't have DNA, but instead they use RNA as their genetic material. And when they enter the cell, as shown here, then they can then unwind. And this unwinding, after that, the RNA is getting translated by the ribosomes, the host cell ribosomes. Interestingly, the newly made viral proteins, which are not many, it's about 2 to 4 viral proteins, they will reorganize the cell, both the membrane structures and also many pathways. And as you can see here, RNA virus replication starts where the RNA is copied into new progeny. And this is a very efficient process, and one viral RNA can produce as many as thousands new progenies. And after that, the virus builds this, when they build it, this replication organelle, then what happens is that the virus will get encapsulated and leave the cell and infect new cells or new hosts. And what is interesting that all this process, although it's very robust, it's very difficult to study because it is very intrinsically connected to the cell. A lot of processes will be changed. So the virus is, during its replication cycle, has to build a very interesting structure, which is called the replication organelle. This replication organelle, I'm showing you this for different viruses, human viruses, insect viruses, and plant viruses. These organelles are built from membranes by the virus using host resources. But as I mentioned, this is currently unknown how, what kind of proteins and lipids are participating in this process. So my laboratory is involved in dissecting the role of different viral and host proteins, how this process, the replication organelle formation, is conducted in the infected cells. The system what we are using is thrombus viruses. These are very small plant viruses. As shown here is encapsulated form. The protein is outside and the viral RNA is inside. Just one single RNA, which is about 5,000 nucleotides. So it's a very tiny RNA. It's about 2 to 3 messenger RNA in length. And it can code for 5 proteins only. So only 5 proteins. As I'm showing here, 2 of them are the P33, which is an RNA chaperone, and the P92, which is produced through a so-called read-through mechanism, where the ribosome in 5% of cycles, it does not stop here to produce the P33, but instead it's read-through and producing the larger P92, which is an RNA-independent RNA polymerase. This is the polymerase which makes the RNA copies. And in one cycle, this virus can produce as many as 1 million progeny per cell. And the other proteins are produced later during the infection, which are the coat protein, which encapsulates the viral RNA, and then the cell-to-cell movement protein, and P19, which is a suppressor of RNA silencing, which is important to fight against the host. So it is important that this plant virus is very similar to flaviviruses and other human and animal viruses as well. So it can be used as a model system not only for plant viruses, but also for other RNA viruses. And here I'm showing you the structures what the virus is building. This is in collaboration with the Cristina Risco laboratory in Madrid. So now this is inside the cell. You can see that this is the yellow is the membrane of the host cell, and the blue ones is what structures the virus is building. These are the so-called replication vesicles. The viral RNA and the viral replication enzyme is inside these blue entities. And the virus builds hundreds of these inside the cell. And it's important for us to understand how this is being formed, because then we might be able to interfere the formation of these structures, and then we can fight against the virus. Why these structures are important is because they are allowing the virus to sequester all the components what it requires for replication in a very small place, which is very good for efficient biochemistry. Also, this is a protecting entity where the virus can hide from the host recognition inside these blue entities, and also it might be able to avoid the destruction by the host, because the host has a lot of antiviral tools, ribonucleases and proteases, which can destroy the virus if the virus would stay unprotected. However, within this small entity, the virus is protected from recognition and destruction. So we would like to understand how this is being formed. And now this is shown here in that how many of these are forming inside the cell. This is inside the cell is the peroxisome membrane, and the virus is facing, as shown here, the cytosol, this small blue things facing the cytosol, but they have only a very narrow opening called the neck structure. And you can see here the gold ones are the ER membrane structure. So the peroxisome and the ER forms a very close entity inside the cells. This is driven by the viral proteins. To understand how these structures form, we have to perform systems biology approach. This is basically to identify all the host components which are participating in virus replication. And interestingly for us, we were able to develop yeast as a system for us. So instead of the plants, the first things what we are doing is that we are replicating the virus in yeast. This is a major technical breakthrough for us that we were able to do this, because in yeast we can do much easier and faster and cheaper genetic studies. So as you can see, and the virus is basically replicating inside the yeast cells very efficiently as in the plant cells. And so it's reaching ribosomal RNA level, so it becomes the most abundant RNA after 24 hours of replication. So very robust process. And now using the systems biology, my laboratory is studying both the performing genomic screens, where we delete or study the libraries where the yeast genes are deleted one by one, and we ask the question what happens to virus replication. Also, we perform proteomic screens to see interactions between the viral proteins as well as the host protein, what host proteins are interacting with the virus. And finally, we are also performing lipidomic screens. In this situation, and we want to understand what's the role of the lipids, because the formation of these vesicle-like structures require a lot of membrane rearrangements, which also will depend on lipids. So after performing all those screens, we want to identify those factors which are pro-viral, so helping the virus to take over the cell, regulation of the cellular functions, and for replication. We also identify factors which are fighting the virus. So they would, if they recognize the virus, they would destroy the virus. These are so-called cell-intrinsic restriction factors. And finally, we also look at factors which would affect the evolution of the virus, where the virus can change and the host components can affect how fast the virus would change to adapt to the cell. After identification of all these factors, we want to understand their functions, the mechanism how these host proteins interact with the virus. And of course, the ultimate goal is to obtain the antiviral approaches to make sure that the virus is, that we can fight against the virus. So during our process, this is a summary. The current status is that we perform many genomic screens in yeast with deletion libraries as well as temperature-sensitive libraries or overexpression libraries. And we also perform proteomic screens with protein arrays and mass spectrometry and so on to identify all the interactors with the viral protein or viral RNA. And again, the lipidomics as well. So altogether, we found 500 host genes, at least at this point, which affect virus replication. So this is a large number, and we are studying several of them. And here it's shown those which we already characterized and published. These are including lipid biosynthesis genes, translation factors, and so on. And those are here which are affecting negatively the virus, so they are fighting the virus infection. Today, I would like to, in the beginning, I would like to talk about kind of a summary of our results and then showing some new ones where I will go to more details. In the summary section, I would like to first to talk about the recruitment and pre-assembly of the viral replication compartment or replication organelle. As shown here, the P92 RNA-dependent RNA polymerase and the P33 protein, after that translation, they would bind to the viral RNA and they recruit the viral RNA into membranes. And these are inside the cell, these membranes. In this case, it's the peroxisome and the ER membrane which is being involved. And the virus also, these replication proteins also recruit not just the viral RNA, but it starts assembling host proteins stolen. These are selected host proteins stolen from the host cell. These include translation factors, protein chaperones, and helicases, and so on. So these proteins will assemble together with the viral RNA at the site of replication. And the other thing what is happening is that to have also the virus requires a lot of new lipids, new membranes, and also using old membrane, already existing membranes from the cell. So the virus will, with its replication proteins, will form so-called membrane contact sites, where two membranes comes together very close. Again, this all happening inside the cell. In this case, one of the membrane is the peroxisome membrane, and the other is the ER, the endoplasmic reticulum membrane. These membranes come together with the help of actin filaments, as shown here schematically, and you can see the electron microscopic image. So now these two membranes are so close, but they don't fuse. So they don't merge together, they don't fuse, they just stay together so close. This is so close that one single protein can bridge the distance between the two membranes. And this allows the virus to enrich membranes and proteins at the target membrane, the peroxisome. This is important to make new membranes, new lipid composition for the replication site. So the virus can actively manipulate the membrane structure. And for this, the virus is using sterile binding proteins, also so-called wet proteins, which are anchoring the virus replication proteins at the ER membrane, and also actin filaments. So, but after rearranging the membranes, enriching lipids at the membrane contact sites, the virus needs to do further work. And this is to deform the membranes into these vesicle-like structures, which are very tiny. The size of these about 70 nanometer, so it's not visible for confocal microscopy, only for the ER. So you can see here I'm showing an electron microscopic image where you can see the round shape. In this round shape structure, the viral polymerase, the RNA-dependent RNA polymerase, synthesizing the viral RNA. First the minor strand RNA, and then lot of new plus strands. And those are getting repelled through this very tiny opening, the next structure, to the cytosol to start new infection. So the virus is building hundreds and thousands of these kind of structures, and my laboratory discovered that to build these kind of structures, the virus is recruiting a cellular factors, so-called escort family. This escort family is a large group of proteins which can deform membranes into this kind of structures. So the virus is recruiting these escort proteins to build these compartments, and hundreds of them. And when we deleted these yeast genes, now you can see that the structure is on the left side is open. The virus now cannot form this closed, semi-closed environment. And when this happens, now the virus is completely unprotected from the cellular virus-degrading enzymes. So the virus cannot replicate efficiently anymore because it will get destroyed by the host antiviral machinery. So this is very critical for virus replication. Here now I summarize all the things which we have discovered and published in the last 10 years from my laboratory. So first I talked about the recruitment process, when the virus is taking dozens of host proteins to the site of replication to form a pre-complex. Simultaneously, the virus also rearrange membrane lipids and enrich particle lipids at the site of replication, as shown here using the membrane contact site strategy. And in addition to this, the virus is recruiting the escort proteins to build this semi-closed replication environment which protects the virus from degradation, from host attack. So all these things I summarized, but now I would like to give some further details. And so for that, I would like to introduce the methods, what is available to us. So in addition to, of course, we do have the plant system where we can study, because this is a plant virus, so we can study things in the plant cells as well. But in addition to that, the new systems what we developed, I mentioned first the yeast system, where the replication is very efficient when we launch the virus from plasmids. We also have the cell-free extract system. So in this situation, we lyse the cell, we destroy the cell in a gentle way, and then using the cellular components, we can reconstitute the viral replicase complex in vitro using recombinant and purified viral proteins, P33 and P92, and that will, if we also provide the RNA, can make a complete cycle of replication, making both minus and plus strands, a lot of new plus strands. So this is something similar to what happens to the inside a cell, but now it's in vitro environment, cell-free environment. We also could rebuild the virus replication compartment using artificial lipid vesicles. This allows us to study the role of lipids separately. So first I would like to talk about what kind of lipids the virus is utilizing and how the virus is doing this. Again, this is very critical for the virus because it needs to build this special environment, this replication organelle. So what we found in yeast cells as well in plant cells is that this is a particle phospholipid labeled here with blue, is that the virus is capable of hijacking or recruiting this lipid, particular lipid, to the site of replication. The virus replication is this, the site is within the cell, this green circle. And as you can see, when we test where is this phospholipid is located, is colocalizing. If you merge the images, you can see the colocalization of this particular phospholipid with the virus replication compartment. This is in yeast cells, and the lower panel shows the plant cell. So again, within the plant cell, you can see that a large portion of the plant cell is taken over by the virus, and this area is also enriched with this particular phospholipid. But how this is being done is a very complex process. So my laboratory discovered that the virus is obtaining these lipids and membranes by hijacking the endosome system. The endosome is a transport system inside the cell, a vesicle transport system. As you can see here is that from a plasma membrane and also from the Golgi, trans-Golgi, the early endosomes forms, and this early endosome has many protein sorting and lipid sorting mechanism, and it will also go to late endosomes, or it will either result in a recycling of components, or it will fuse to the vacuole, what is called also lysosome in the mammalian cells, for degradation. So what we found is that the virus is converting this early endosome machinery with the help of Rep5, which is as shown above here. It's an enzyme, it's a protein which can activate different pathways through binding to GTP and binding to effector proteins. And the viral replication proteins bind to this Rep5 and recruiting this to the site of replication. And again, the major goal for the virus is to build these elaborate structures, very light structures, using these extra lipids and extra membranes to do so. So one of the critical enzymes for this, what the virus is hijacking, is the so-called VPS34, which is a PI3 kinase. This kinase is, and here is shown the structure of it. You can see the membranes, and this is the protein complex, including the VPS34, the kinase, and this is a lipid kinase. So it will produce from a phospholipid, it's phosphorylated and make it into a PI3P. This is important for endocytic trafficking, for so-called autophagy, and also for trafficking, lysosomal trafficking. And what we found is that when this enzyme is deleted from yeast, this is now shown here. So comparing to the wild type situation where the yeast has all the genes, you can see very robust replication. This is an Orden blot showing how much RNA is accumulating. So you see that a lot of RNA is accumulating in the control samples. However, when we deleted this gene, VPS34, now you can see that the replication is very poor. However, when we express from plasmid, because the origin of the deletion is in the yeast chromosome, but now from the plasmid we supply the VPS34, the Y-type one, we can now obtain even higher level of replication when this enzyme is abundant. However, when the enzyme is a mutated form, which does not have the kinase activity anymore, and we supply this protein to the system, to yeast, now this cannot complement virus replication. The virus replication is still poor. So this enzymatic activity is required for the virus. And here I'm showing to you that indeed the virus is very robustly recruiting this protein to the site of replication. The blue shows the confocal image inside the cell. That this is where the virus is replicating, the aggregated peroxisomal system. And you can see the VPS34 localization. Most of the VPS34 is now colocalized and with the peroxisome marker. This is an aggregated peroxisome. The virus is inducing the aggregation of and proliferation of these structures. And you can see the very nice colocalization, although some VPS34 still available to the cell. So not all the proteins are stolen by the virus, but a significant portion of it. This is also shown the same in the plant cell, not just the previous was one in yeast. And so you can see that these interactions, the replication protein will now colocalize with the Rep5. Again, this is the endosomal protein what the virus targets. And you can see also the lipid, which is being made by the kinase. These are all colocalized inside the cell. So basically the virus is stealing away this system and this lipid from the cell. And when we delete this Rep5 protein similar to the kinase protein, now this is basically three different genes in yeast, and these all Rep5 proteins. But when we delete all three of them, now what we can see that the virus replication is very poor inside the yeast cells. For this, we use this tomato bushy stunt virus, as I mentioned earlier. This is a peroxisome replicating virus. Fortunately for us, we have another Tombus virus, which is carnation Italian ring spot virus, which is using a different subcellular organelle, so-called mitochondria. So this allows us to study virus replication in different environments, subcellular environment inside the cell. So when we have this second Tombus virus, the mitochondria replicating virus, you can also see that deletion of these Rep5 genes cause major problem for virus replication. So not depending on where the virus replicates, now the virus has to recruit these Rep5 proteins and VPS34 kinase to build its replication organelle. And this is also shown here with the peroxisomes. You can see the enrichment of phospholipid and Rep5 and the virus replication proteins, all these together inside the plant cell. In this situation, it is the peroxisome membrane, and for the second Tombus virus, it is the mitochondrial membrane. So the virus is very efficient with taking away these components from the cell for its own purpose. And when we delete this Rep5 from the cell, now the virus cannot steal the phospholipids anymore. So you can see the localization of the phospholipids, which is enriched when in the Y type is the virus is replicating. However, when these Rep5 genes are deleted, now the phospholipids are not enriched at the site where the virus would like to replicate, and the replication is very poor. So in summary, this first part, you can see that the virus is taking away the endosomal system with the help of Rep5 to recruit to either the peroxisome or for another virus to the mitochondria inside the cell, and to build these large replication organelles, which is very unique to these RNA viruses. Another very interesting process which we are studying currently is that how the virus obtain energy to support virus replication. All these processes are very robust, happening in 16 to 24 hours. There are very dramatic changes inside the cell. How the virus finds energy to do this is what my laboratory discovered with the help of those screens I mentioned earlier. Our laboratory discovered that the virus is also hijacking glycolytic enzymes. These glycolytic enzymes inside the cell are a very ancient process. It's based on utilizing glucose, which is the major energy source for many cell types. So this glucose, the sugar, is being converted to pyruvate inside the cytosol of the cell to obtain different compounds, chemical compounds, as well as, importantly, ATP. There are two steps. You can see here the PGK1 produces from ADP ATP, which is a very high energy containing molecule. And also the pyruvate kinase, which is CDC19 in yeast, this is the name, also producing from ADP ATP. This is basically the energy currency inside the cell, where these ATPs can be used for many processes. They are fueling a lot of process by providing energy. And what we found is that the virus is very efficiently hijacking this ATP-producing enzyme from the cells. So here is the PGK. When we co-express, we purify the virus replication components, the entire complex from the cell, and we look at it, what other proteins are co-purified with this complex. And this Western blot shows that the PGK1, this ATP-producing enzyme, is very efficiently co-purified with the virus replicase complex. So the virus is taking away this from the cell. This is also shown here in confocal images, where you can see that the virus replication compartment and the PGK1 is very nicely colocalized. Not all, but many of the PGK enzymes are now part of the replication compartment. When the virus is not present, you can see that PGK is distributed in the whole cytosol. Similar things happening in the yeast cells with the second ATP generating enzyme, the pyruvate kinase. Now this is colocalized with the virus replication compartment during the infection. So this shows that the virus is very efficiently hijacking the enzymes. And when we study this in vitro, in the cell-free extract, we can demonstrate that indeed this is directly happening. So now we are assembling the virus replication complex in vitro using yeast cell-free extract. So the origin of the yeast does not have any viral components. We provide that purified components from bacteria, where we propagate them in bacteria. And as you can see, when we use a yeast mutant where we can downregulate the expression of PGK1, now virus replication is very poor when PGK1 is present in a very low amount. So this is a direct evidence that PGK1 is very important for virus replication. Similarly, when we study the role of the second ATP generating enzyme, the pyruvate kinase, and now we are using a temperature sensitive mutant in yeast. So when we elevate the temperature during yeast growth, this enzyme is partially inactivated. You can see that virus replication is in big trouble. So the virus probably cannot obtain enough ATP for its replication. And also this is shown in plant cells that the virus is very actively taking away the pyruvate kinase from the cell to the site of replication. And when we, in plants, to confirm the data what we obtained in yeast, when we downregulate, we silence the expression of these proteins, now you can see that the plant is very healthy. You can, healthy from not being infected by the virus. You can see here that we cannot easily detect the virus replication. In the wild-type plants, in these plants now the virus is very actively replicating, as you can see here with node blot, very high level of replication, and the plants will be killed very shortly, as you can see here, the dying plants. But again, when we downregulate these ATP generating enzymes, now the virus cannot do the same to the plant. It cannot replicate to high level at all. To demonstrate directly that indeed the virus is utilizing this for ATP generation, we adopted a system where we can detect ATP generation. In this situation, there's a bacterial protein subunit which has two conformations. In one conformation, it has this extended structure, and in another conformation, it has this compact structure. So now, technically, we are using a system where we have two fluorescent proteins. These two fluorescent proteins can make, during confocal microscopy, we can detect their excitation state. So when it's ATP-free situation, when a low level of ATP is around, then this subunit, this ATP binding subunit is in this extended form. In this situation, when we use light to illuminate only the CFP, now this cannot pass the energy, the light, to the Venus, the yellow fluorescent protein. So in this situation, now when we detect the yellow excitations, we find very low signals. However, when ATP-bound form, in this situation now there's a rearrangement of this protein which binds to the ATP, and now this brings very close vicinity these two proteins. And so if we excite with laser the CFP, this can easily pass the energy to the Venus. So when we detect using this light channel, we can detect now this fluorescence, and this is called FRET. So what we've done, we connect this so-called ATP sensor. We connect it with the virus replication protein, we fuse them together, so now we measure what's happening in the vicinity of the viral polymerase. Utilizing this system, now what I would like to show it here is different images in yeast cells, is that where the virus replication protein is, that this is the scale here. So when it's very intense, whitish and red, now you can see that the FRET signal is very high. So now it means that it's an ATP-bound form. So there's a huge amount of ATP being produced at the site of replication, in the vicinity of the viral polymerase. Now, when we use the mutant enzyme, this is a temperature-sensitive pyruvate kinase in the yeast, and this is the only copy available for yeast, and we use higher temperature. Now you can see that the signal is shifting towards the blue, which would mean that now the viral polymerase in an ATP-pure environment. There is not much ATP around the polymerase. So overall, this suggests that indeed hijacking of the pyruvate kinase and this ATP-generating enzyme from the glycolysis is important for the virus to enrich, to obtain ATP at the site of replication. And now I'm going to show you time course experiments to show that indeed this ATP is being utilized by the virus. So this is a very early 4-hour time point after we express the viral proteins. And now this is no replication, this is the pre-assembly process. And as you can see now, the ATP sensor is measuring very high ATP levels in the yeast cells around the viral polymerase. At the 8-hour time point, when replication starts very intensively to produce viral RNA progeny, now you can see that the signal is getting more bluish, meaning that the ATP is now getting used up by the virus. And by the 12 hours, when the replication is really intense, you can see now the signal is the ATP signal is now very bluish. So the virus is intensively using up the ATP generated by these glycolytic enzymes. The same is happening in the plant cells. So when we express these components, viral components, during infection in the plant cells, you can see also that here very high FRET signals can be seen on the right image. So the virus, not only in yeast, but also in plant cells, is doing the same thing. And biochemically, to show you why this is important for the virus, this is a biochemical assay. The virus is during replication producing a double-stranded RNA. So originally the virus is single-stranded, the plus strand, and the polymerase would make it into a double-strand RNA. And this double-strand RNA then has to be unwound to obtain structures which is the polymerase can utilize. So here we artificially make a probe which will hybridize. It's a reductive probe hybridized to portion of the viral RNA, and then we provide enzymes which the virus is stealing from the cell. These enzymes are helicases. These helicases can unwind the structure of this double-stranded RNA structure. So you can see here the double-stranded RNA moves into the J like this. But when we provide the pyruvate kinase together with its substrate, now it provides ATP enough, which this liquid host proteins can use to unwind and obtain single-stranded RNA, which is suitable for virus replication. This is just one example. We do other assays to demonstrate why the recruitment of the ATP-generating enzymes is so important for the virus. And this is summarized here. And so a lot of steps from the pre-assembly of all these host proteins and also the replication, as I showed with the helicases, all this requires ATP. So the virus is hijacking these ATP-generating enzymes to provide ATP locally, and basically using the host cell energy system to obtain energy for its own replication. So these are the different steps and the different enzymes involved in summary. So to summarize our presentation, my presentation today, the virus, the tomato bushy stunt virus co-ops conserve cellular factors. These factors are also hijacked by other viruses, although their details usually are not known. But what we are very happy to see is that our virus is utilizing these conserved factors also used by the cell and also by other viruses during their infection. And so these include different translation factors, stress components. It also involves glycolytic enzymes to generate energy. It also endosomal compartments, as I previously mentioned, from lipid kinase to Rep5 regulating proteins, and also the actin cytoskeleton. All these are the virus is hijacking to build these elaborate structures to support replication, very robust replication. And again, our ultimate goal is to interfere with these processes to help the cell survive infection. However, there are still critical questions unanswered. For example, what co-opted host factors are stolen by the virus only for short period of times, and which are those which are permanently staying inside the replicase complex? Those would be primary targets for antiviral approaches. And also, what are those factors which serves as assembly hubs? Currently, our thinking is that the virus is not recruiting all the components one by one, but it will recruit many things through complexes, already pre-existing cellular complexes, to make the process faster. Finally, I would like to thank all my collaborators and the PhD students and the postdocs during the work, including Judy Pogani, who developed the system for cell-free studies, and Tadas Panavas, who developed the yeast system, and Kai Zhu, who developed the cell-free, the liposome-based replication system, as well as Chinkai Chuang for the glycolytic work, as well as Dr. Kovalev for his work on the helicases. And also, I would like to thank the funding agencies, the National Science Foundation and NIH, for their funding, and especially the collaboration with Christina Risco with the structures of the viral replication compartments, and with Andy White, who collaborated with us on the role of the RNA and protein interactions during replication. And I thank you for listening to the presentation, and I'm ready to take questions. Thank you.
Thank you, Dr. Nagy, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of your presentation window, type in those questions in the box that appear on your screen, and click on the send button. We'll answer as many questions as we have time for. So let's get started. We have a great audience today. Our first question is, why do viruses need to induce membrane proliferation and deformation during infections?
So the virus basically has accomplished two major roles by this. The first one is to build its replication compartment, where it collects all the host proteins and all the factors required for replication. This is important for biochemistry, that it will make everything very robust by collecting them into a smaller environment. And the second one is that the virus it's easy to recognize the invading virus because it's so robustly replicating. So the host cell can launch an antiviral response to destroy the virus. This is a built-in mechanism in the host cell. So the virus is building these cellular compartments protected by these lipids. These lipid membranes is basically to hide away from the recognition by the host cell. So the host cell will not easily recognize that it's being utilized by the virus because the virus is hiding in these compartments, pretending that it looks like a cellular membrane structure. Thank you. I'm ready to take the next question.
Thank you, Dr. Nagy. Our second question from an audience member is, what is the advantage to be an RNA virus versus a DNA virus?
So the different genomes, DNA genome versus RNA genome, is a very interesting question. And based on studies, it looks like the advantage of the RNA viruses are very common, and they are the most abundant in eukaryotic cells versus the bacteria, the cells, the prokaryotic cells. The RNA viruses probably has advantage because they can very quickly replicate in the cytosol. So most DNA viruses will need to enter the nucleus, so they have to go through not only the outside membrane, the plasma membrane, which protects the cell from the outside environment. Viruses need to enter and go through this protective membrane. So the RNA viruses only have to go through, most of them only have to go through this outside the plasma membrane, and then they can right away start replication of their RNA genome. So it's very fast and relatively easy process. DNA viruses, on the other hand, they have to not only go through, and most of the DNA viruses not only have to go through the plasma membrane, but they also have to enter the nucleus where DNA replication occurs inside the cell. This is the natural, the cellular replication. And so it requires basically going through multiple membranes and utilizing the cellular DNA synthesizing machinery. And this is a more challenge for DNA viruses, so they are replicating usually much slower than RNA viruses. So eukaryotes probably favor the RNA viruses are more common because they are just faster, more evolved for the eukaryotic cells than DNA viruses. Thank you.
Thank you, Dr. Nagy, and thank you for our very engaged audience today. We have quite a few questions coming in and have time for a couple more. How much cell damage is caused by the virus infection?
So viruses, to build these so many progenies and to rearrange membranes and all these processes, it can make a lot of damages to the cell. So it really depends on how much particle virus is adopted to that host cell. Most of the time, when the virus is very well adopted to the cell, the damage caused might be not enough to kill the cell. So the virus can, again, when its adaptation is very good between the virus and the host, then both can work together or at least coexist. However, when the adaptation is not as good from the host cell side or the virus side, then many times the virus can cause so many damage that the cell will be killed. So in different environments, the different interactions, it will be decided. Different hosts, different viruses have different reactions, which can go from very severe lethal damage to a little bit less damages as well. So a lot of different responses can happen. Thank you. I'm ready to take next question.
Thank you, Dr. Nagy. And we have time for one more question today. Our last audience question is, can other viruses replicate in yeast? Interesting question.
Thank you. Yeast is a very good host system because unlike other eukaryotes, especially plants or more developed mammalian systems, the gene redundancy is very high and very high number of genes these eukaryotes have. So yeast is much simpler system, so you can study in yeast things much faster, easier, and much cheaper than in other eukaryotic cells. So yeast is an excellent model system. However, this is a major challenge how yeast can be utilized, developed for virus research. So currently the number of viruses which can be replicated in yeast is growing. In addition to the thrombus viruses I presented today, there's also originally developed by Paul Alquist's laboratory is the bromosaic virus, which is another plant virus, and also an insect virus, which nodaviruses, which can replicate in yeast. But now the numbers are growing, and one of my former postdocs developed a DNA virus replication system in yeast. So the numbers growing, but it's still relatively low, but it's a major advantage. But it's important that after we discover things in yeast, we have to go back to the original plant system to confirm those findings. Thank you. I finished my answering today. Thank you.
Thank you, Dr. Nagy, so much for your wonderful answers. Do you have any closing remarks you'd like to share with the audience before we close our webinar today?
So I really enjoyed this possibility. I really enjoyed this opportunity to talk about viruses and virus-host interactions today is one of the most exciting area of research, because in addition, of course, that viruses are very dangerous plant and animal and human pathogens. They can cause a lot of harm to human health, as well as, of course, in agriculture, they do a lot of damages. So it's important for us to understand and protect ourselves and our environment from viruses. But the interaction between viruses and the host is much more complex. There are examples where viruses have positive effects on different systems. There are also cases when we utilize viruses to, in medicine, to obtain ways of killing pathogenic bacteria, or utilizing them for curing different diseases, including cancer. And viruses are also important through their interactions is to understand what causes disease in different organisms. viruses are easier to use as intracellular pathogens to understand what is the disease and what is health for a cell and for an organism. And also through the pathogenic process, the interaction process, we can understand functions of the cellular proteins. So different cellular functions. We can develop new ways not only for diagnostics, but also for curing different diseases with chemicals. So these are all things which the virus-host interaction studies can help us. So I'm very excited about continuing this research, and many of my colleagues share this vision as well. Thank you so much.
Dr. Nagy, thank you so much for your presentation and for your important research. I'd also like to thank our audience members and especially LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 2017. You'll receive an email from LabRoots letting you know when the webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thanks for joining us, and we hope to see you again. Bye-bye.